So we're not going to do that. So it's 60, 60, 40. That's why it's the 4 points individual, 6 points team. Okay. This question here is one that really is something I'd like you just to work through on your own. The best bet would be to work through each of those sentences and just pick out the mistakes. Okay, so find what's wrong in each of them, and there's only one that's all the way correct. Okay, so mark and count the errors in each, and with all of them, you're starting at the radial styloid. Okay, I'll give you a hint just so you know. So some of you will probably be emailing me and asking, but A is the one that's correct. Okay, but go through and look at all of those and figure out which one, or just figure out where the mistakes are. That'll help you get familiar with those anatomical directions. Okay, so you will need to know those anatomical directions and the planes for exam 1, even though that material was mostly presented in lab, also a bit in this TBL stuff as well. Okay, good. So yeah, so remember you're sending in number 6 on your app there. Let's see what most of you said. Good. So, all right, everybody's back. Okay, welcome back. So you can see on the screen there, I just kind of filled in what's going on with each one. So really the key thing here is to remember those key enzymes. Okay, so the first step there, that helicase enzyme unwinds the DNA, pulls the 2 strands apart. Okay, and then we see in step 2, the primase enzyme lays down RNA primers onto the DNA first. Okay, so those are the first 2 steps, right? And yeah, again, if you don't get all this right now, because we don't have a ton of time, you can come back to this recording and pause here and fill that in if you weren't able to fill that in yourself. Okay, this one here, step 3, we have our third enzyme that's a part of this process. So this one's DNA polymerase. Okay, this adds complementary DNA bases now. Okay, so removes those RNA primers, brings in the right DNA bases to make that new strand of DNA. And you can see that there's a new strand of DNA being formed on both sides of the old DNA, right? So then we end up with 2, and each of those 2 strands now, each of those 2 double helices, those each have 1 new strand and 1 old strand, okay? And that's just a term called semi-conservative replication, okay? All right, so yeah, so that's that. So we're going to keep moving on. And what we'll do, I probably won't finish all of these, so I'll just pause the recording as everybody's leaving, and then I'll finish up these slides and add those to the end of the recording, and I'll post that video. So if you want to hop back on there and check that later, you can. Okay, so a couple of things here that we'll just talk about briefly. These 2 different processes that are a part of this section, DNA replication, right? The point is to, obviously, to make more DNA. Okay, more DNA, okay? And this happens before cell division, right? So before a cell divides, we need to make it so we have 2 full sets of DNA, so each cell has the complete DNA, okay? So that's what DNA replication is all about. Protein synthesis is using, there's no E in using, using the DNA to make proteins. All right, which is really the purpose of that DNA, is to be the kind of the code for protein manufacturing, and we'll walk through that. So they're similar. Obviously, they both use DNA. They both use some RNA in the process, but the end goal is very different. Okay. All right, so we're going to take a few minutes here. We'll wrap up with this just to kind of follow the steps that a protein would go through. And so there's obviously many different types of proteins in the body. Some of our hormones are proteins. And so insulin is an example of a protein hormone. Insulin is the hormone that allows for glucose to enter into cells. Okay, it's responsible for binding to cells, allowing glucose to enter, so that glucose can be metabolized and form ATP, our cellular energy. Okay, so that means it goes through this series of steps through what's known as the endomembrane during its formation. Okay, so these next few questions will kind of walk us through that. Okay, so go ahead and let's do this one here on the clicker app. Okay, so what's the first step in the formation of insulin? And so the 2 key things to remember with each of these really is where in the cell is it happening? Okay, so which organelle, and then what's happening, right? What's being formed. So go ahead and choose A through E there. We'll go quick on these, give you about 15 seconds from here. Oops, sorry about that. Okay, a couple more seconds. Go ahead and send something in. If you're not sure, doesn't matter. You don't get graded for being right on these. Just get graded for submitting something. Okay, I think that's close to everybody there. Go ahead and show what you said. Okay, so most of you are saying D. Okay, so the first step is transcription of messenger RNA in the nucleus. Okay, those are the three key things there. Where is it happening? The nucleus. What's the process called? Transcription. And what is the goal of the process? To make RNA, right? Good. Okay, so that's step number one. Okay, after RNA is made, after transcription is over, what happens next? And we can just kind of walk through this. Remember, that's why that's one of the reasons we have those nuclear pores. Okay, so it's going to go out of the nucleus, and then which organelle comes next? Okay, so it moves out of the nucleus, and the next one is the rough endoplasmic reticulum. Okay, so it moves out of the nucleus into the rough ER. Okay, so that's the correct one there, B. Okay, so we'll keep moving on. After it gets to the rough ER, what happens? Okay, I'm going to go ahead and open this one up on the app here. So go ahead and answer that one. Okay, go ahead and send in your answer when you're ready. All right, anybody else? Give you about 2 seconds. There we go, thank you. Okay, so what did you guys say on that one? Okay, so about 3/4 of you are saying C. So the messenger RNA calls for the correct amino acids to form the polypeptide chain. Good. Okay. And yeah, so next step here is polypeptides form. So now we have that polypeptide chain. First of all, let's go back, and this is called translation, right? That's an important piece.
Dr. Sawyer, I have a question.
Yeah, sure.
So I put A, the mRNA calls for the correct nucleotides to form the polypeptide chain. Would that be wrong, like as an answer on the test?
Yes, that would be wrong. Yeah. Because
Okay,
it's not calling for RNA.
Because I'm just thinking nucleotides.
It's calling for
okay.
amino acids.
I just read the nucleotides and not amino. Okay. Gotcha.
Yeah, and it's actually not nucleotides anymore. Now it's amino acids. All right, so it's not RNA or DNA it's calling for at this point. We're taking those triplets and going to that table, right, like we would, and then get like you did for that question, and then you're pulling in the amino acids based on that. Okay, cool. All right, and that process is called translation. Okay, so next step, once we've formed that polypeptide chain in the rough ER, by the ribosomes, right? The ribosomes are the protein manufacturing machines. They're not organelles, but those are the ones doing the work in the rough ER. Okay, so what happens next? Where is it sent next? And that's where it's sent to the Golgi apparatus, right? And the two things that happen in the Golgi apparatus, typically processing and packaging, and really it's going to be kind of sent to the right spot in the cell, right? So it could be used right there in that cell. It could be sent out of the cell via exocytosis, or it could be just stored in a transport vesicle for a while until it's needed in the cell. So we kind of think of the Golgi apparatus as kind of the post office where packaged, shipped out. Okay. All right. And then, yeah, and then the final product, insulin in its final form is protein hormone. What's next? Okay. Yeah, so next step after it's formed, and this is what I kind of just said, we got a few possibilities for what could happen, right? So it could be stored in a transport vesicle until it's needed. So that would be like those beta cells in your pancreas that actually make the insulin. They're making insulin all the time, so it's there ready for that to be used when needed. C is also true. It transfers to a transport vesicle that buds off the Golgi apparatus. That's true. Okay, and then D, it can be immediately transported to the surface of the cell to be exocytosed out. Okay, so all three of those things are true in that answer. And so those are the steps of protein synthesis. Okay, and this is kind of showing those steps together. First step, messenger RNA production. All right, second step, synthesis of early insulin and then transport into the Golgi apparatus. That's second, third step. Fourth step, final processing and packaging in the Golgi apparatus. Lastly, either stored in vesicles. And then finally, if it's something like insulin that needs to go out of the cell, it's exocytosed when the body calls for it. Okay. All right, and this is just what insulin does. You definitely don't need to know all the details of this picture, but here's insulin binding to a receptor on a cell, causes this whole chain of events, which eventually causes this protein called GLUT4 to come to the surface, and that allows glucose to enter the cell. Okay, we'll talk more about this when we talk about diabetes next semester. Both type 1 and type 2 diabetes have connections here. Okay, so we won't do this right now, but this is a very good piece of practice for you to do. Okay, so you're gonna write in the DNA bases here, right? That would match up with this, and then take this one and write in the RNA that would match up with that. So the RNAs are gonna match with the DNA there, and then take these two sets, take these two triplets of RNA, go up here to the table, figure out which amino acids. There's one amino acid there, two there. Okay, so there's another example like this in your book. You want to practice that multiple times, okay, before the exam, because that'll be something that you do. Okay, why don't I just do this so you have this on the video, and then you'll be able to quiz yourself. Okay, guys, we went a little bit over. Thanks for hanging in there with me. Again, I'll be hanging here for a sec. Hey, by the 10:30 class, I wanted to do just a quick video walking you through this slide because I felt bad rushing through that too rapidly. Okay, so this is something you're going to need to know how to do. So let's walk through it. Okay, so typically the question will be something like this, where I give you either 3 DNA bases or 6, and say we could start from there and just say what are the amino acids that are going to come out of this, or we could first say which DNA pairs, which DNA bases match up with this. It's complementary base pairs, and then from there we could say which mRNA do we get out of this. Okay, so it's all, whenever you have 2 DNA strands, we're always going to need to tell you which of the 2 that you're going to use to make the messenger RNA, right? Because either one could be used. Okay, so let's start here. So I give you this first set of 6 bases of DNA. Okay, so we got 3 A's and then an ACT. Okay, so we're just matching DNA first. So the first thing we're going to do is DNA. Okay, so the ones that match with the A's are the T's. Okay, so we got T, T, and T. So you got 4 A's in a row, so you get 4 T's in a row. Next, the ones that match with the C is a G. Okay, and then lastly, the one that matches with a T or complements the T is the A. Okay, so let's see if I'm right. Good, got it. All right, so that's what we got there. Okay, so now the instructions say use this one, right? So we're using this one to make our messenger RNA. So we need to be able to do complementary bases that match this DNA strand here. Okay, and remember, we don't have we have to use the U here instead of the T. Okay, so the T doesn't exist in DNA, so we got to use the U instead. Okay, so the ones that match up with the T are going to be A's, right? So those are just like the DNA, no difference there. Okay, so we got 4 A's in a row that are going to match with this T, this T, this T, this T. Okay, so we got our RNA there. And then next is going to be the G. What matches with G in RNA is still C. Okay, and then again, what would match with A, if it was DNA, it would be T. We already did that one. But since this is RNA, we don't have the T, so we got to use the U instead. Okay, so we got that. Okay, so A, C, U. Okay, now we're going to take this, the messenger RNA, we're going to take 3 bases at a time. We're going to take this up here, and we're going to figure out which amino acid each one calls for. Okay, so the first we're going to start with 3A. So you start over here with the first base is an A, second base is an A, and then we're just going straight down. Oops, sorry. And we're going straight down, and we're looking for another A. So we find that one here, right? That gives us lysine, L-Y-S. Okay, second triplet codon is ACU. So again, we got A for our first one, but this time it's C for our second one, and then U for the third one. So we're going ACU right there. That calls for threonine THR. Okay, so there we go. So that's how we would do that. Okay, practice that, use some other ones, come up with some of your own, practice, use them. That'd be a great way to learn that. All right, guys.